Yo, and welcome to Shooting the Soil. Today we are looking at compost from Catalyst BioAmendments. And so far we're starting right up here at the top of the slide. And look at all the fungal hyphae we see. I've been quite excited to check this out. I've been holding off to enjoy the experience with you all live. But I got a good feeling from what I've heard. They're Dr. Elaine Ingham students. That's super cool. And this is what I just got shipped. So already we're seeing fungal hyphae all over the place. We're at 100x right now. Just kind of scanning around. Getting an idea of what we're looking at. Seeing if we see anything cool. Hopefully gonna find some nematodes in here. But yeah. Ooh, that's... looks like we got some fungal spores in there. Kind of interesting looking. Why don't we dive into 400x real quick and just take a look at that. Oh yeah. We got full manual mode going on right now, so we got to adjust our settings. Get a little bit more light, a little bit more focus. There we go. And where were we looking? Ooh, that's probably a big fungal spore right there too, but where were the little ones that I wanted to see? Oh no, lost them. Actually, now that we're down in here, we could get a look at the bacteria, and holy cow, a lot of bacteria. All sorts of life wiggling around down there. See a lot of coccyx, and over here we got some bacillus shape, another bacillus shape, basilisk, and then we got these more round ones, which are the more common. It almost looks snaky, but I think that's more basilisk. Same with that one. I think it's quite a spirilla, but looking pretty good. We could actually go a little bit more light. There we go. Ooh, that's an interesting little something split in there. I believe that's most likely a yeast. Kind of looks like that. Dividing away. Or could be pretty small for an amoeba. Well, let's see what else we can see down here. Well, that really dark thing's looks like some kind of spore. Oh, we can see a nice fungal hyphae right over it. Clear, very young, but good septa. Those little black separations. And pretty uniform, despite where it's all mashed up right there. Alright, let's go back into about that focus range. But yeah, look how much life is in this compost. More fungal spores. Almost thought that was a amoeba shape right there, but nope. I think that's... yeah, that... actually, this... I believe that would be I think that's an amoeba right there. Get a little bit more centered. Another good sign. What do we got wiggling around? Flagellate of some sort? Little cilia? Yeah. Nice to see. Two more strands. But yeah, feel free to ask any questions in the chat. You're checking out and giving you guys an idea of what a good compost should look like. Oh, we see another amoeba right there. I wonder what that is. That's interesting looking. That might be, actually, I kind of think that's a nematode in a cyst or an egg. Baby nematode? Good. It's my best guess. Looks like there's a little organism hanging out in there. And it kind of looks like a little nematode. Lots of amoebas. Never was able to find. Oh, that look at big old segment, little piece of a nice looking beneficial fungi. What else do we see? Another amoeba. That looks like a little testate amoeba. Another one with a really dark spore there. Ooh, we see some nice fungal hyphae in this aggregate. Oh yeah, very nice looking fungi. Oh, it keeps going. See the separations. Nice coloration. Keeps going. And keeps going. Alright, it goes into that aggregate there. Think it comes out the other side? Oh, wow. And that's an interesting spore there. It's got some ribs on it. It's a big spore. Let's see if we can find where this fungal hyphae goes. Kind of lose it under that aggregate there. Does it come out the other side anywhere? Nope, but that's pretty good. Oh, another one going this way. How far do you go? Nope, only right there. This one's a little bit younger. Hasn't got that quite brown color. Hasn't filled up with nutrients yet. But that's a good sign that it's fully alive and growing. Oh, what's bumping around into that? But, wow. So much life in there. Yeah. You can't say that all this isn't doing in there. When we take one drop out of a 1 to 5 dilution, out of 1 millimeter of soil, and we see all this life in just a little piece of the slide. Like, we could keep scrolling. Yeah, I'll just give you an idea at 400x how long it takes to scroll all the way to the bottom of the slide. Yeah, we're way out of focus now. More spores. We're still going. More life. And there we go. Now we're to the bottom. Or just about. Yeah, there's just a tiny bit more. But yeah. That is a lot of nature's workers. More fungi. Oh, it's interesting looking. It's just a little curled piece of organic matter. But then the small one's a fungal strand. What's this over here? Alright. Let's go out to... We're going to go back to 100x. We're going to get a little less light. There we go. Focus back in. And... Alright. Let's see where we're at. Wow. Another big... Really big fungal hyphae. Ooh, I think it continues even more up here. Wow, that's an even bigger one than we just were looking at. Holy cow. I need to find a nice fungal hyphae that's shaped like a perfect S to update my logo. I need a Shooting the Soil S fungal hyphae. Alright, but yeah, look. Every single... Yeah, this is an over... Like, whoa, there's a ton of bacteria, so it's probably pretty close to a 1-to-1 bacteria to fungal ratio. But there is fungi everywhere. Alright, let's find a nematode. Actually, we could, anybody see anything moving down there? Just checking out that aggregate. Oh. There's a nice little amoeba there, I believe. Another one. Those are aerobic. What are those? Arcilia. I was just researching them the other day, and I forgot how to exactly pronounce the name. But wow, fungal hyphae everywhere. So cool. Yeah, getting this compost is definitely a good way. Oh look, we got two of these amoeba actually splitting right there and multiplying. Very cool. That's probably another fungal spore. A lot of diverse spores. A lot of hyphae. Yeah, but this is definitely what you want to see when you look at your compost under a microscope. Let's see if we can find a nematode in here. They did say they were experiencing a very wet period when I got the compost. But yeah, you would expect for if it got too wet or something like that to start seeing a lot of ciliates, and we are yeah, we're not really seeing too many ciliates. Usually if you put compost and do a tea, you start seeing a lot of ciliates for sure. Oh, something's moving around there. What's that? Let's take a closer look at this. Switching over to 400x. We need some different light. I have a Swift 380T. It's a pretty decent, inexpensive microscope. Can't really complain. I do want to upgrade, but that's just because I'm a... Oh no, I'm hitting the slide. I went too far. Back up. Fail. Alright, let's try to get refocused here. Where am I at? Am I too close? I lost my focal range. We're getting close now. There we are. Alright, I knocked everything around. And probably that organism that was moving stuff around is long gone because I whacked the objective right into the slide. Yep, and now I got no idea with what was moving that around. Oh, but we found another nice amoeba. Alright, where... Nope. I think we're going to have to go back out. What? Little fungal spore. I like the football-shaped ones. Interesting piece of something. Organic matter, I believe. But let's go back out to 100. Readjust. refocus without banging the objective into the slide. And let's scan around a little bit more. See if we can find something moving in there. Yeah. A lot of nice aggregation. All sorts of organic matter breaking down. It's a good sign. Let's see. Spotting more amoebas and all sorts of good stuff in there. Go over a little bit this way. Actually, let's have some fun and switch over to the dark field. Maybe that will help us find some nematodes a little bit better. Alright, change some settings here. Make sure we're all the way up. We got our light all the way open. There we go, we're getting close. Oh, more light. Alright. And now we're doing dark field microscopy. Yeah, good day, guys. Checking out some Catalyst BioAmendments Compost here. And we just switched over to dark field. Just trying to do a scan around. And wow, look at all that bacteria sparkle back there. Looking at that big glowing thing, but yep. Let's see. Want to go a little bit back over this way. But yeah, it is fascinating. Looking at stuff in the dark field. This would be closer to what the organisms would see, or actually really gotta get epifluorescence to really see closer to what they could possibly see. Yep. Take a little focus on this nice organic matter in there. Getting all broken down. Alright. Hopefully you can find something cool to look at swimming around down there. But yeah, looking really good so far. How's it going, Micro? Yeah, there's a lot of life in here, a lot of diversity. Over a little bit more. Surprised we haven't spotted a nematode yet. But yeah, all sorts of good. It looks like some kind of cyst there, and maybe looks like another amoeba. Amoeba there, I think. All sorts of good facultative aerobic organisms. All we're missing is a nematode. Saw a flagellate. Lots of amoeba. We always like finding the nematodes. Sometimes it takes a couple of drops to find them. Sometimes I'll go through 2 slides, not find any, and then find like 4 on the next one. It is funny like that. I always feel it's like just my luck. But yeah, definitely a cool way to do my microscopy in the dark field. Nice and easy to spot the amoebas. They kind of glow a little bit differently, especially if you move the light around. Yeah, see, now they're getting a little neon glow over there. Usually phosphoresce a little bit. All the living stuff does. Alright, let's see. Almost just thought I saw something moving. Yeah, they definitely can, like, go dormant or seem pretty good, but I was doing a couple of experiments where I took the same sample and like left it out and put one in the fridge, and it was much harder to find the nematodes after I put it in the fridge for a couple of days and then thawed it back out. But after just leaving it, putting it in a plastic baggie, leaving it for a couple of days, just a small amount of soil, I was able to see very similar results, surprisingly. So I wouldn't think shipping would be that big of a concern. But the nematodes were the one thing that I noticed was the difference after freezing. I think they do kind of just go dormant. Oh, more amoebas. So we could be just scanning right over them, not seeing them because they're not wiggling about right now. Yeah. Take one last scan across. But yeah. All sorts of life in this compost. Very excited to add all this biology into my compost pile, onto my plants. Yeah, I did an extract and put this on this morning. Just a small little extract of the small pouch and a small net, and just added in some water, stirred it around, and put it right on the plants. And so far, they seem to be loving it. Oh, that's a big old hyphae. It's cool how it glows under the dark field. The hyphae look totally different in dark field than they do in the bright field. Yeah, not too shabby. Yeah, still hoping to spot a nematode or something in there, but see a squiggly thing, but I think it's a fungal hyphae. Not a nematode. Interesting looking piece of plant matter. A nice big fungal hyphae glowing right there. Definitely one of the best composts I have looked at yet. Look at this hyphae. It's like glowing orange, the outsides. It's pretty cool. A little bit better focus. That's about as good as we're gonna get. Alright, scan around just a little bit more. But yeah, probably gonna wrap things up pretty soon here, so feel free before we end to ask any questions or pick my brain about regenerative soil. Be happy to help, or about anything microscopy. Yes. Definitely pretty good looking stuff. Definitely a whole lot of fungal hyphae everywhere. Little tiny strains. Because we're out at 100x right now in the dark field. Fortunately, that's all I can get dialed in with this microscope. Hoping to upgrade in the future. But yeah, that's probably a fungal spore. I can tell from the way that it's just glowing on the outside. It's probably alive. It's interesting how the life does that. Really starts glowing, and that's why I want epifluorescence, because it will do that even more. Then different wavelengths will make different stuff glow. It's pretty neat. I just used the remote software that came with my Sony camera. So yeah, it works. It was just worked really well and convenient, and actually, yeah, gets a pretty good image. So I haven't really experimented with any of the like the actual microscope camera and what they come with software. But I pretty much sure any remote camera software should be pretty sufficient. It almost looks like a. Sure if that's a really, really big hyphae or not. Like that's definitely a fungal hyphae right under it. Yeah, let's switch over to bright field. Yeah, actually, so I don't blind you guys, let's turn the ISO back down to there before we switch over. Pull out the little plastic dark field filter, and then we need less light. Yeah, that kind of. Yeah, it looks pretty hyphae-like. It's interesting. It's actually. We gotta go into 400x and take a closer look. I want to see bacteria inside a fungal hyphae. Or even inside a root would be even cooler. Alright. Where was that strand? There it is. Alright. What do we see? Do we see anything in there? Yeah, so we got obvious fungal hyphae over here. But what is this? Is this also a hyphae? And what is this circular thing in here? Just a different type of hyphae. It looks like, kind of looks like nutrients in there, or like a humic acid. You wouldn't think a root would be that uniform. You think it would taper off. So look over here. Huh. Yeah, I would say that's just a large hyphae. I'm taking Matt Powers' course right now. Yeah, it is. I always forget if it's the fulvic or the humic acids that create that nice brown color. It's one of those living acids. Still about halfway through the course right now. Looks like we got a little flagellate I'm trying to focus on in the middle there, but it's moving up and down too quick. But yeah, that's quite interesting looking. Alright, actually we gotta put a little shade on things. Get just a little bit more light. There we go. That looks a little bit better. Yeah. Another cool amoeba. Arcella. Yeah. Arcella. I'm pretty sure that's how you say it. Arcella. That's an Arcella amoeba. Another fungal spore. But yeah, it's just when you scan around in here at 400x, it's like you just see things everywhere. That's when you know you got a good compost. All different shapes, a little wiggly bacteria down there. Nice organic matter. The little fungal hyphae growing out of it. All helping for that reduction. Making things plant available. Yep. Another Arcella. Oxygen liking amoeba. Aerobic. Alright. More hyphae. Ooh. Little spiky piece might be a piece of a nematode or a what is it? Larva of a nematode. Let's actually learn about that. Oh. Nematode larvae look like little spikes in the soil. I always thought it would be like a, little bug leg or bug antenna that broke off in there or something. But I think a lot of it's actually nematode larvae. Just thought I saw something moving over here, but another good example of fungal. I don't know. I think this is definitely one of the most fungi I have seen in a compost. That's very cool. Very excited. Even though we didn't find a nematode, I bet if we looked around in here long enough, we'd probably find one. Or I'll find one in the next drop. Oh, there's a classic little hot air balloon testate amoeba. That's what I think they look like. You got the balloon, and then you got this flat part where the air would go up. That's how I remember them. Alright. A lot of cool looking stuff. Probably scan around and look at this all day, but we gotta wrap up the stream here at some point. Oh, there's a really nice dark brown fungal hyphae. Alright, well, I think that ought to do. Hope you liked the experience and learned a little bit about, what we should be looking for when we look at our compost. And yeah, hope you got some new goals, things to strive for. And yeah. See, someone just saying hi, so if you got any questions, we were just about to wrap things up here. Looking at this really nice looking compost. Oh, there's another fungal spore. Ooh, fungal hyphae and a spore. Wonder what the little hexagon shaped spores are. That's definitely a spore. Ooh, I just saw a little fragilate move quick up there somewhere. Where'd he go? Yeah, Diego, that's one thing I really want to do, but I need the better scope with the top lighting to really see through the root. But smaller root hairs you can kind of do with this scope, but that's the main reason I want to upgrade to a scope with epifluorescence and the ability to top light and bottom light my subjects, and that's going to make it a lot more fun to looking at roots and seeing bacteria and fungi actually inside of the root. Oh, there goes it. There's a little flagellate bumbling around like a bumblebee. Where are you going? Ah. Funny little creatures bouncing off stuff. Alrighty. Well, that ought to do things. Yeah. Like, subscribe, share, help the channel grow. Would be much appreciated if you would be so kind. And until next time, we will catch you on the next one, and happy growing. Good night, guys.